Hi everyone, my name is Daartey. I am currently a PhD candidate at St. John's University in Queens, New York. I work in a cancer research lab that is focused on childhood cancers, such as neuroblastoma. And don't let the name deceive you, neuroblastoma is not in the brain. It develops on the adrenal gland that sits on the kidney. Neuroblastoma is diagnosed in children less than 5 years old. Just imagine the lifespan of these kids diagnosed with cancer, their quality of life, and their development. Devastating. So, currently we have treatments that are available combined with radiation therapy, and also there are emerging immune therapies that are available. However, the problem is that despite these intensive therapies, neuroblastoma still evade standard chemotherapy. And our lab has characterized specific cancer stem cells that are able to evade or avoid standard chemotherapy treatment, as well as radiation therapy. And this subpopulation of neuroblastoma cancer cells is about 1% of the tumor bulk, which means that if you use standard chemotherapy to kill the neuroblastoma cells in the patient, the 1% that is left is not sensitive to the standard chemotherapy. So as a result, we are looking for the mechanisms. We are trying to investigate the mechanisms and also find targetable or druggable avenues in order to cure or kill these cancer stem cells. And currently, we are exploring the PI3K-mTOR pathway. This is normally functional in cell survival and also important for cells' metabolic processes. So, what we have found so interesting is that we use a small molecule inhibitor that blocks this pathway. And by doing a series of experiments, we found that actually when you block the PI3K-mTOR pathway, it leads to activation of other pathways that compensate, and this still enhances the survival of these cancer stem cells. So we have done advanced molecular biology experiments such as qPCR. We have also done RNA sequencing, and we have identified these pathways that we can target. So in the future, we are looking at combining this small molecule inhibitor that targets the PI3K-mTOR pathway with a standard chemotherapy, and also this compensatory pathway that has been activated with targeting the PI3K-mTOR pathway. We'll do this in the in vitro in the cell culture plates, and I'll also do this in the animal model to see how effective this will be in curing neuroblastoma in kids. Thank you so much for listening to me, and I hope you enjoy my presentation. I am more than happy to receive any questions you may have. Thank you.